Okay, I think we can start on the differential expression session. And I've created these slides together with Olga Detlefsen in SciLife Lab. And she also got some of these slides from Åsa that you met yesterday. And learning objectives will first introduce the basic concepts, where we will define what is differential expression, talk a little bit about what's typical for the single-cell RNA sequencing data compared to the more traditional bulk data, and then look at some of the most common distributions that are used. And then later we will talk about some of the common methods. And you will later, at the end, do a rehearsal where we use those methods. And then we will show the performance of these methods using a couple of comparative studies. And we will also try to look at all the results simultaneously at the end to find common genes that are significant using all the methods. So what is differential expression testing? You're probably quite familiar with it. So what one is doing is to take the read counts, usually have a count matrix, and then you want to compare between two experimental groups. And now we will assume that you have two different groups. You can also do this similar thing here between cells. So for example, if you infer cell types, you can find differentially expressed genes between cell types. But as this cell type identification will be more talked about tomorrow, we will not talk so much about this now. But so we assume that we have two groups that we will compare. And then for these two groups, you observe some counts, and then you will try to assess whether these counts are different between the two groups. And this differential expression is an old problem. It's known from both bulk RNA-seq and microarrays. And it's testing between groups is an even more old problem in statistics. So comparing and finding group differences is quite known. And there are two of my pictures that are not showing. It might show if you have Safari browser, but not in other browsers. So I'll. Sorry? Yeah.
Sorry? You like click and view image, then show.
Ah. Wait. New tab.
Yeah, that's my show one.
Oh, excellent. Okay, yeah. I agree. Thanks. So I try to show the difference between these count matrices from the bulk and from the single cell. So what you have in the bulk RNA-seq is that you have n samples, and these could be from two groups. Quite often one are comparing two groups. But what's important here is that in that first sample, it's not just from one cell, but it's from a mix of cells. And these cells could be, of course, from different cell types. And what happens when you have such a mix of cells is often that you get averaged out the counts. So between the different cell samples, you'll get quite similar counts. You might have differences, more differences between sample groups, of course, but they are often quite similar. At least if you compare to the single cell, because here the columns are from individual cells. And this might vary a lot because they might be from different cell types. So I just made some numbers here, but trying to show that you have much more variation, and quite often you have this what is called dropout. That for this isoform here, you actually have no expression, or at least not any detected expression. So this means that the distribution, so we are trying to fit the distribution for each isoform or each gene. And this distribution here from the bulk will be quite different from this single cell, because you have much more variation often in the single cell. I'll go back to... And some of these characteristics of single-cell RNA-seq data are mentioned previously. You have a high noise levels, both technical and biological factors. You have low library sizes. So for each cell, you don't have that many reads as for the bulk. And you have this low amount of available mRNAs that result in amplification biases and dropout events. The 3' bias, partial coverage, and uneven depth. I think Åsa mentioned the stochastic nature of transcription. So some of these characteristics are technical and some are biological. And quite important is this multimodality, which I'll show example over here. And this is because you have various cell types, right? So, for example, for this one, showing a bimodal distribution, it might be that this peak here is representing one cell type or a collection of cell types, and this peak represents other cell types. But as you see, so these are real data, picked out some genes, and it's very difficult to find one distribution here that will fit all the genes. So we have combinations of, or you have a lot of variation in the distributions. So not just unimodal or bimodal, but you might have even more modes. And often you have this high peak around 0, because many of the reads are 0. But in some cases, you also don't have that many zeros. So it's very hard to really model this data. So let's go over to the methods. So this is an example, only some of the methods that have been developed to test if genes are differentially expressed. You see a list here. So this was from a publication by Wang and others in 2019. And you have also more examples shown in this other publication here. So these are 2 comparative studies that I will be talking a bit more about. We can divide the methods in some main classes. You have, first of all, you have non-parametric tests, and you have parametric tests. So if you're looking at non-parametric tests, they could, for example, be Wilcoxon rank sum test, Driscoll-Wallis, Kolmogorov-Smirnov. They are often based on ranks, and some of them fail in the presence of a large number of tide values, which is the case here because you have a lot of these zeros. Parametric tests are usually more powerful, but that's only if the distribution that you assume for the parametric method is close to the truth. But we saw all these different distributions, so it's quite often that it's not possible to find some known statistical distribution that will fit all the genes. So therefore, it might be better to use non-parametric methods. But I'll show the comparisons of all these methods in a while. And then you can also not just divide on parametric and non-parametric, but you can divide upon those methods that were built for bulk, like this famous EDGER and DSEQ2. And then you have some that were made specifically for single-cell RNA-seq. And I just mentioned a few of them here, and you probably know some of them from before. Yeah, and then we have these parametric distributions that are used. If you model, you can either do a non-parametric fit, or you can use some model, some statistical distribution. And here I mentioned some of the most used ones. The negative binomial, which has been used a lot for the bulk RNA-seq. But due to this dropout effects in single-cell RNA-seq, so you have a peak around 0, you have some other distributions that might fit better, like the zero-inflated negative binomial. And you have also combinations of the Poisson and the negative binomial, which might fit better than the negative binomial. So just a little bit statistics. What you will do when you have two groups that you will want to compare is that these two groups might, here's just an histogram, so you can think of this as read counts, a count for each cell. And it might look like this if you are in a special situation where the data are actually normal distributed, which they usually are not for the single cell. But if we had something like this, then you run a regular Ttest between the two groups using a T-test statistics like here, where you assess whether the difference is different, taking the difference between, test if the difference in mean is different between the two groups, and then divide upon the estimated standard deviation for the mean difference. But yeah, actually there have been some methods showing that using a T-test could be okay. And a reason for this is because of this central limit theorem, which you might have heard about, saying that if you have a large number of, when n becomes large, number of samples or number of cells, then everything goes towards a normal distribution. So, so that might also hold for the cell counts or the read counts within a cell. It goes towards a normal distribution. So the T-test might be quite good actually. But of course, it will not take into account these dropouts. So the normal distribution will not fit the dropouts very well. So what is usually done is to model the gene expression and assuming some random error. You estimate the model parameters. You use the model for prediction and or inference. And often what we're talking about here is to infer whether a gene is differentially expressed or not. And the better the model fits the data, the better statistics will be. So it's quite important also for the power of a test to use a distribution that fits the data well. I'll just show some examples of the most common methods. Here is the negative binomial distribution. It has two parameters. The first one is the mean. And if it only has this parameter, or this alpha is set to 0, then we have actually a Poisson distribution. So therefore the negative binomial can be seen as a generalization of the Poisson. So it has this extra parameter here called the dispersion parameter. And if you increase the dispersion parameter, you'll get a distribution which is wider. So here it is almost 0, and we have something quite similar to the Poisson. You see the mean value is 4 here, so we have 4 counts. And if you increase it a little bit, you see a little bit change in the distribution. But if you increase it, this dispersion parameter to 1, then we get something like this. So even though the mean value is 4, you quite often get 0 counts. And in a few cases, you get quite a lot of counts. Like 25 you can actually get. And you see that here you will get something which is more like what we will have for the single-cell RNA-seq data, because we have this a large number of 0 counts. So this is why this negative binomial could be quite okay for the single-cell RNA-seq data. And the mean values is this mu, and the variance is mu, which is equal to the Poisson. But then it's actually it's an error here. It should be times and not divided upon. It's times this alpha parameters. So this is quadratic in the mean value. So when you get high mean values, so for example, a gene that has 1,000 transcripts, then this variation becomes extremely high. And therefore, often not just one common alpha is used, but instead an alpha that is either a combination of the variation in all genes, a combination of that and what is shown in that gene, or it can be estimated in a gene-wise manner. And actually, if you have enough data, it's probably better to do it gene by gene, because genes might have different variation. They are not very similar. So here's this zero-inflated negative binomial. It's quite similar to the negative binomial, but you have you can also include a dropout parameter, essentially saying how large percentage of the cells that will have a dropout. So you can see how it is affected by going from 20% dropouts to 50%. And on the last one, I've just increased the mean value. So this is starting to get more similar to what we will have in a single-cell RNA-seq experiment. I'll just mention briefly one more distribution. It's a Poisson beta, which is actually also a generalization of the Poisson. So in this Poisson distribution, you have a lambda parameter, which is the mean value. But if you allow this mean value to also vary a bit, and it can vary according to a beta distribution, then you can allow for more variation. And depending on the parameters in this beta, you can have different shapes of this Poisson beta distribution. So this is also able to catch this dropout effect of increasingly many zero counts. And I think it's on in this paper here, or this method here, BPSC, where they use this Poisson beta, and they actually have an interpretation of the parameters here, which are rate of activation of transcription, rate of inhibition of transcription, and this last parameter, G, is the rate of transcript production while transcription is active at the locus. Okay, then I don't think I will spend too much time or any time at all to go through all these methods. You will can read about them when you do the exercises, because there are a lot of methods and new methods are all the time coming. And but they often use these distributions that I mentioned in various ways. And a few of the newer methods are more non-parametric. Yeah.
I was wondering, so we were talking a bit before this, that's a natural step for you to go from after clustering your data is to perform differential expression between the clusters.
Yeah.
I was wondering if what's in general terms, how small can a cluster be for you to still get something out of the differential expression analysis? Because I imagine that this is a bigger problem than it is from standard bulk RNA-seq analysis.
Maybe some are more experienced to say something with that.
So one of, I mean, there are many approaches to that, because then that is a case of unbalanced groups, right? So you're comparing 5 cells against 5,000 of the rest.
Yeah.
So in some of the packages, it's possible also to, for example, filter the cells. So for example, each group should have at least 100 or at maximum 100 cells. So at least the groups are comparable to each other.
Yeah.
So that would be one approach.
But what I was asking this specifically because even for groups with 5 samples, if you have so many zeros, it may be a problem or not.
Maybe you're not grouping with 5 samples. Maybe you had expected to have a little bit more.
Yes.
Yeah, but then if you have that number of samples, let's say, usually a cluster would have maybe 50. I would say I consider a real cluster around about 50, especially for a single cell. Then you have 50 against maybe a maximum of 100 or 200 if you limit that group to the rest. So for single cell, common practice is that you identify your clusters and you want to find markers. So also that is implemented in the Surat package that you can. So the statistics is based. You compare this cluster against all the rest of the cells. So if you have a big data set, let's say 500 cells versus 20,000, then it's quite a big difference in cell number, right? So in that case, you also increase it or reduce the p-value just because you're increasing the amount of observations. So that's another way that you can do is randomly select the cells from the rest group and compare that to the same amount of numbers from your cluster. But there are different alternatives to that. Some of those methods also correct for it, so you can actually include all the cells.
Okay, so I'm going to talk about some comparisons of these methods using 2 comparative studies. But first, some more basic about errors, which is more general statistics. But it's maybe easier to grasp it if you think now about what we're dealing with which are the differentially expressed genes. So here you have, to the left, we have all those that, so here are the true positives. This means those that are truly differentially expressed. So to the left, you have all those that are truly differentially expressed, and some you will say that are significant, which will be true positives. Oh, sorry. Let me see. And the other ones are false negatives. So what you'll get here is the sensitivity is those truly differentially expressed genes among all those that are in fact those significant genes among all those that are truly differentially expressed. And to the right, you have the specificity, which are those that are not called differentially expressed among all that are truly not differentially expressed. So sensitivity, so that's the main thing we will talk about now, is this sensitivity, which is also called the true positive rate. Also you have the specificity, which is also called the true negative rate. And it's often trying to balance out these two. So the best methods will both be sensitive, really finding those that are truly differentially expressed, but also not saying that those that are not differentially expressed, not saying that they are. So if you are able to find a method which is having a sensitivity of almost 1 and also a specificity of almost 1, then you have a very good method. The first comparative study is from Wang and others, came this year. So they are using both simulated data and real data in order to assess the performance of all these methods. And in the simulations, they are using different scenarios. The first scenario here, so these are the two groups that we will compare. The first scenario is the unimodal scenario. So both groups have a unimodal distribution, but they are not having the same mode. So that's why they are differentially. So this gene is differentially expressed. I think it will be significant. And it's actually a real gene that shows a distribution like this. So actually this figure is not simulated. It's from a real data. But they will use this scenario later when they simulate some data. Another scenario showed by another gene is when you actually have bimodal distribution for both groups, but it's just the proportion that are in the different groups that are different. So, for this group, we see that most genes are in the first peak, and the other reads here are in the smaller peak, but it's a different proportion here for the other groups. There are more reads that are having this high counts. So that's another way that two groups can be different in. For the next scenario, you actually have 2 peaks for the first group and just 1 peak for the other. And this peak is on the same place, so they have the same mode for that peak, but, except that this other group doesn't have this bimodal distribution. And then you have another type here where this group just have 1 peak, but the other group has 2 peaks, and also this peaks are not at the same place. So yeah, there could be also other possibilities for genes to be differentially expressed, but at least these are some that also are found in the data. And then they also simulate some distributions for those genes that are not differentially expressed. So what they do is to simulate some genes that are differentially expressed using all these scenarios, and then also assume some that are not differentially expressed using these distributions here for those ones. And then you can calculate, because you know on this simulated data which ones are the true positives, the true negatives, false positives and false negatives, and so on. And you can calculate for all cutoffs, you can calculate specificities and sensitivities. So cutoffs here means that you can vary the significance level. Often you use the false discovery rate. And then you look at the whole range of possible cutoffs. And then for all cutoffs, you calculate both the sensitivity and specificity. Then you can draw lines and calculate the area under the curve, which is a quite common method or measure to evaluate whether classifiers are good. And you see here that you can get a ranking of the different methods. Yeah, you can just note that this one, SIG-EMD and DE single, are on the top of the list, whereas some quite well-known methods for single-cell RNA-seq, like SCDE and monocle, they are having a lower AUC. Then you can also look at these different scenarios and see if you see any patterns here. In general, it's more difficult if you have not just this unimodal, but if you have different modes, it might be more difficult for the methods to get good positive rates. One thing which is quite important here is the effect of sample size. Because if a method is good, then it should, if you have enough samples, so samples here are cells, the various measures should go towards, so the true positive rate should go towards 1, and the false positive rate should not, should also go towards 0. Accuracy should go towards 1, and so on. And you see that for most methods, it actually helps, or for all methods, it helps to have a lot of samples, but not all methods are going very fast towards what you want here. And it seems actually for this method here, it seems to not be enough with the 400, and you will not, even if you increase the number of cells, you will not get an accuracy of 1. So these were based on simulated data. And usually it's better to use real data to compare methods, but the problem then is that you usually don't know the truth. So if you have some data, you don't know which genes are truly differentially expressed. But actually, you can use some data which might give some clues about it. For example, this data set here from 2011, they have verified 1,000 of the differentially expressed genes. Or the 1,000 were verified by real-time PCR. And in this data, we'll use those as a gold standard. So these are the truly differentially expressed genes. In order to find false positives, one could do resampling. So even though we have two groups here, so one group was from 48 mouse embryonic stem cells, and 44 were from mouse embryonic fibroblasts. If you then make groups where you take just at random, some from this group and some from this group, then you should not get any significant genes. So the significant genes then will be the false positives. So then for the first measure, the sensitivity, where you use this these genes that were verified to be differentially expressed by a real-time PCR. You can look at how many of those did you call significant, and that will be the sensitivity. You can note here that for quite a lot of the methods, you have a quite low sensitivity actually. So it could be around 20% and so on. So it varies quite a lot. But what is important to note here is that you can get as sensitive result as you want if you just include, say that almost all genes were significant. So you also see here that the more genes that were included, that were called significant, the higher sensitivity you will get. But of course, you will get a lower specificity there. And this is illustrated in this figure here. This bar shows the number of genes that were called significant. But the blue ones are those that were intersecting with those 1,000 reference genes. So the goal here should be to have a high, have a large blue part here without having the yellow one too large. And then for the false positive rate, so actually in this using this resampling of the groups or of the samples and say that they belong to other groups than they really belong to, you will generate data sets where you should get no significant results. So this should be, you should get number of detected differentially expressed genes should be 0. But for some methods, so they are in general quite good at this. But for some methods, you get some significant genes, even though. And then you can calculate the false positive rate. Then they went a little bit further in this comparative study to look at what happens on the more pathway level or functional level, where they looked at both KEGG and gene ontology terms, and looked at how many such terms are found. Of course, even though you find some terms here, they might not be the correct ones. So these numbers don't tell too much. But they had some experts in the field that knew what was expected from this data, which, yeah, so these were stem cell data. And these experts were asked whether this found gene ontology and KEGG pathways, if they were, how relevant they were. And then you can get an overall score for each of the methods. And actually we saw those methods that might not show so good results for the individual genes, they actually got higher score. And we were looking at these gene ontology groups and the pathways. So it's not easy to draw an overall conclusion from these results. So, it's a bit small. So, in general, the more sensitive a method is, the lower the specificity gets. So, it's no method that really tries to optimize both these two. We saw that tools developed for single-cell RNA-seq tools that are developed for single-cell RNA-seq data, they focus on handling zero counts or multimodality, but not both. And it has been shown that these methods that can capture multimodality perform better than the methods that are designed to handle zero counts. So that's at least something they state in this paper. But in general, there are no clear winners here. So two of these methods that were more non-parametric and designed for single-cell RNA-seq was this DE single. So it was one of them performed quite well. So a couple, and this SIG, what was it called? SIG-EDAM was also performing quite well. But one has to keep in mind that the first author of this paper, the comparative study, was also the first author of this this method. Always something to keep in mind when we are reading comparative studies. And we don't see clearly that these methods for single-cell RNA-seq are much better than the bulk RNA-seq methods. And one can conclude that since both that the methods show very different genes, and I didn't show that actually, but when you compare between methods, it's not much overlap. And also that the methods are not perfect, and there are no methods that are much better than the other ones. One could claim that more method development should be done in order to get even better results. I really try to find methods that capture this special characteristics of the single-cell RNA-seq data. I'll just briefly mention the other comparative study, which also find much of the same, at least this. So the higher on top here, they have also taken speed into account. So these are a lot of measures, both sensitivity, specificity, and so on, accuracy, and also how long time it takes to run the method. You see actually this edge-R is developed for the bulk is on the top here. And many of these methods made specifically for the single cell are at the bottom actually for this comparative study. But I think in general it's having a method that are able to capture various distributions is probably the way to go. Either using a parametric distribution that can do that or use a more non-parametric approach. Any questions before we start to go over to the I just show at the end here. Hopefully you will get plots like this at the end, where you can plot for each gene difference between the two groups. Because even though the methods are fancy and should be able to give reliable results, my experience, at least with the bulk, is that when you start plotting, you can see that the ones on the top of the list actually might be an outlier or so that just make two groups different. So it's always good to really plot the individual counts between the two groups and see if do I really trust these results. So that's one thing that you should learn from the rehearsals that you will do soon, and also that you can try to combine the various methods and find those genes that are significant in many of them that will give you more robust results. So that you are also asked to do in the rehearsal. So I think we can start the rehearsals and we will go around and try to help.